All right. Hi, my name is Shaun Stice, and I'm a senior scientist here at PathogenDx. Today I will be presenting PhytoX. It's a novel multiplexed assay for the rapid detection of 9 plant viruses and viroids of concern in cannabis sativa. Here we go. So, some of the major drivers of yield loss in a majority of crops are weeds, which can reduce yields, especially in a field setting, insects, which can cause damage to fruits and lower yields as well, and then of course plant diseases. In cannabis production, weeds are not of primary concern because most cannabis production is done in a highly regulated environment, usually intensive production that's done indoors. And due to this, high amount of care that is taken for these plants, weeds are not really an issue for indoor growing operations. However, insects and plant diseases can be a problem. And the major issues that growers may see are viruses and some insects, especially mites and thrips and insects of that nature. So for this talk, we'll be specifically focusing on viruses, and viruses kind of fall under that category of plant diseases. I also want to mention that insects play an important role with many viruses. They are able to spread viruses from plant to plant, and so controlling insect populations is really important for virus control, especially if there's an outdoor component to your growing operation. So the causative agents of plant disease fall under several groups of organisms. So first of all, what is a plant disease? A plant disease is a continuous irritation that's caused by a biological organism, and that results in the decline of the plant and altering the plant's physiology. Some of the major groups include fungi, bacteria, viruses and viroids, protozoa, nematodes, which are small parasitic worms that feed on the roots of plants, and also parasitic plants. And these are kind of an interesting group, but they're not really a huge issue on cannabis. So for the main problems we see on cannabis are usually fungi diseases and then viruses and viroids. Now for fungi, it's actually pretty simple to see the physical fungal structures on the plant. So you have symptoms and signs. So a symptom is what the plant looks like, the plant's kind of response to the pathogen. And then the sign is the physical presence of that pathogen on the plant. So with fungal diseases, you can actually see the hyphae. If you use a small hand lens, you can also often see the fruiting body structures under a microscope or often with the naked eye. So diagnosing these types of fungal diseases is relatively straightforward and doesn't always require the use of complex molecular tools. However, for viruses and viroids, you often do need molecular tools to confirm a diagnosis. The viral diagnosis, again, it requires molecular tools and it can be quite challenging. And the main challenge is that these symptoms can be quite variable depending on the plant, the genotype of the cultivar you're working with, the environment that the plant's being grown in. And a lot of the symptoms are similar to abiotic factors such as nutrient deprivation or like water stress, things like that can have symptoms that are pretty similar to virus symptoms. So some of the main symptoms you can see in a virus infection in plants includes yield reduction, stunting, mosaic patterns, yellows and chlorosis, ring spots, leaf rolling, growth abnormalities, necrosis, and wilting. So over here I have a couple examples of some of these symptoms. So this is a beautiful kind of ring spot mosaic-like pattern on a peanut leaf. And you can see tomato spotted wilt virus here. So very characteristic of this virus on this plant. And then you have some ring spot symptoms on some peach leaves up here. And this right here is actually a tomato plant, and you see kind of this abnormal leaf curling. So you might think that this is in fact a virus symptom, but actually this is a result of herbicide damage. So this just kind of drives the point home that it's really difficult to diagnose a virus on your hemp or cannabis plant without the use of a molecular tool, because there's so many other factors that can give you symptoms that are similar to the symptoms that you see with the virus. So why is it important to test for virus? Virus management strategies are limited. Unlike fungal organisms and insects, there's not really a spray that you can put on your plant to make the virus go away or to control the reproduction of the virus. It's best really with virus infections to just start out with clean plant materials. And so you want to use these exclusion or avoidance strategies where you try to sequester healthy plants away from virus infected plants. And you can also do some insect control if you have issues with a virus that is vectored by an insect. So some examples of the use of virus testing to limit basically yield issues as a result of virus is surveillance testing. So a grower could test a couple of their plants in their grow occasionally to see if they have any viruses come up. And then if they do have viruses, they can potentially look into different methods, such as destroying plants that are infected or starting out with new clean plant material to avoid having the introduction of viruses in the future. It can also be important to test incoming plants to avoid the introduction of a virus. So if you're getting a new cultivar from a nursery, you may want to test that cultivar to ensure that it does not have viruses and you're not bringing those viruses into your growing operation. And most importantly in cannabis field would be the routine testing of mother plants. So most growers are taking cuttings from mother plants and then propagating or rooting those cuttings for production in their operation. If this mother plant is infected with a virus or viroid like hop latent viroid, all of the cuttings from that mother plant will subsequently be infected, and you can have a dramatic loss in yield sometimes depending on the cultivar. So it's really important to make sure that your mother plants are clean when you're doing a lot of this clonal propagation. There are some methods to eliminate viruses and viroids from plants. Usually they are expensive tissue culture methods where you send off your mother plant and basically they will grow some shoots that have been isolated off of the plant in tissue culture tubes, and they will use something called thermotherapy where they actually grow these plants under high temperature conditions. And the idea is that you will have division of the plant cells that is fast enough to kind of outcompete or avoid the virus. And you'll subsequently isolate a clump of plant cells that are virus-free and then regenerate an entire plant from that tissue that is ideally free of your virus. In a lot of specialty crops in the US, this is done through the National Clean Plant Network, which is a federally funded program to provide virus-free propagation material for a lot of fruit and nut crops. Obviously, due to the regulatory status of cannabis currently, that is not something that is supported nationally. I also want to mention that just because your plant tests negative for a virus doesn't necessarily mean that it is completely virus-free. In order for a plant to be virus-free, you would have to use a technique called RNA-seq analysis, where you would simply isolate the RNA from the plant and do whole genome sequence or next generation sequencing of those transcripts. And then you can actually make sure there's no novel viruses present in your plant material. For most testing methods, we're only looking at viruses that we know the sequence of, whereas with next-gen sequencing, you're able to find, completely novel viruses in your plant as well. There's a lot of factors that can impact how, your plant responds to a virus. And I want to just mention that it's entirely possible for some plants to carry a high virus titer, but still have a relatively good yield. So it's just really up to the grower. If they do test positive for a virus, it's up to that grower to determine, if they believe that this virus is having a negative impact on their operation or not, and what they want to do as far as their decision moving forward. As far as viral threats in Cannabis sativa, we have kind of been focusing on these 9 virus targets for the development of this assay. It includes hop latent viroid, which is relatively well known in this area as causing stunting and dudding in plants, beet curlytop virus, which is primarily an issue for growers with an outdoor growing component to their operation and industrial hemp growers, lettuce chlorosis virus, which has been shown to come up sometimes, especially more in European areas, but potentially over here in the States as well. Cannabis cryptic virus, which has been associated with some grower issues. However, the exact link between symptoms and presence of this virus has not been fully elucidated. Alfalfa mosaic virus, Arabis mosaic virus, tobacco streak virus, tobacco mosaic virus, and cucumber mosaic virus. So these are the associated publications, where they've kind of talked about infections of these viruses in cannabis or industrial hemp. So for our initial round of developing this assay, we focused on developing a multiplex assay for all of these different viral targets. So you can kind of do one test and see, do you have any of these high priority pathogens on your crop or not. Here's an example of symptoms for a couple of those viruses on cannabis. So for hop latent viroid, it's a very small disease-causing particle. It's just a small circular piece of RNA. It's only 256 base pairs in size, but this tiny little piece of RNA is still able to cause dramatic reduction in yield in cannabis plants. You can see here infected cutting with a stunting-like growth pattern. Oftentimes the flowers may not fully form, and this can be, again, it's not great because you've incurred a lot of expenses already trying to grow this plant, and now you're not going to get the yield that you hope for. For lettuce chlorosis virus, you get these chlorotic symptoms on some of the older leaves, and again, this can look like nutrient deprivation, so it's tricky to determine if it's the virus based on the appearance alone. Beet curlytop virus, you get this curling of the smaller leaves, and this is primarily an issue for growers that have an outdoor component to their operation, and this is vectored by insects. So again, insect control, very important, especially, both indoor, but especially in outdoor operations. As far as the current diagnostic tests that are available on the market, they fall into several different categories. So there are LAMP or isothermal loop-mediated amplification platforms, and these platforms usually can only test for a single virus target at a time. They're very useful though, in that they're relatively simple and straightforward to run. However, again, it's only one virus you can test for with a single LAMP assay. Sometimes it can be a bit more, and the sensitivity of LAMP assays is generally a little bit less than that of RT-qPCR assays. So these are those typical like TaqMan based assays, and they can multiplex to maybe 3 or 4 different virus targets, really depending on the assay in question. But again, this still requires you to buy multiple assays for each virus target if you really want to test your sample or your plant for each of the viruses. With the PhytoX RT-PCR microarray, we have an enhanced multiplexing capacity that allows us to test around 10 or more targets. In this case, there's 9 virus targets and 1 internal control, and we have sensitivity that's equivalent to RT-qPCR which is a great plus. And sample time, prep time really varies depending on the method you're using. We have a relatively quick method that we've been able to have great success with for our platform, and that's compatible with some of these other platforms as well. But I just wanted to mention it really varies on the quality of the extraction that you're trying to go for. With next-gen sequencing, you have an unlimited multiplexing capacity because you are literally looking at all the RNA transcripts from your plant. But the sample prep time takes a huge amount of time. Also, this is going to cost a lot of money to do this next-gen sequencing. And the sensitivity can be quite high depending on read depth. I also want to mention there are no antigen tests listed here, so there are ELISA tests and immunostrip tests. Those will work for traditional viruses that have a protein coat. However, hop latent viroid is a naked piece of circular RNA, so it does not have a RNA coat, so antigens cannot bind onto it. Therefore, you can only use these nucleic acid diagnostic tests in order to test for the presence of hop latent viroid. So now I'm going to introduce our microarray technology. So essentially what the PhytoX platform is on PathogenDx's microarray platform. It is an endpoint reverse transcriptase PCR reaction, which is then hybridized onto a microarray. And then the microarray output is read in a computer, and that will determine if you have the virus presence or not. During the PCR reaction, the targets within the virus are amplified and labeled with Cy3 primers, and that's what hybridizes to the array. As far as the development process, we first do an in silico design, and then we do some in vitro testing to confirm that the assay is functioning correctly. To design our assay for each of the different virus targets, we pull down all of the virus sequences from publicly available genomic databases, such as the NCBI, and we then map those to our reference genomes, and we look for regions of high coverage to ensure that our assay is going to work with as many virus strains that have been sequenced as possible. This is an example of cucumber mosaic virus, for which there are 4,000 sequences available. However, with some of these other viruses, such as cannabis cryptic virus, there's not a ton of sequencing information available. And so that makes it a little bit more challenging to know if the test is going to work with every single strain. So it's important to, moving forward, especially within the cannabis community, if there are positive hits for any of these viruses, to hopefully publish them and submit them publicly to the NCBI so that everyone can improve their assays moving forward. As far as the PhytoX process flow, this is generally how it works. Customers or growers will select a plant that they want to test. They will take samples from a couple of those leaves. It will then go to the lab for testing. In the lab, they will conduct a simple sample prep, which I will go into, and they will then take this extracted sample and add it to a RT-PCR tube with the reaction components, run that in their thermocycler, and then the reaction will subsequently be hybridized onto the 96-well microarray. After the hybridization process, it will be imaged and ran through our automated analysis pipeline to interpret the results. Here's an example of what the bottom of the 96-well plate looks like. So we have several different things here. One is a probe. This is a negative control probe. So ideally it's not going to bind to our targets. And this just tells us kind of the background level of hybridization that's occurring. The next circle is the internal control. So in this case, we're using a beta tubulin gene in the cannabis plants. So this tells us if the PCR reaction is working correctly, and it also tells us if the lab technician correctly loaded the sample into the well. And last is our virus target. So in this case, this is hop latent viroid, and you can see there's a signal here in this HLVD positive sample, and the internal control is off as well. This is an HLVD negative sample, so we're not seeing hybridization of those HLVD probes. and we also are not seeing a signal for our negative control. These are Psi3 channel signals. So again, this is what is in our primers that are in that PCR reaction, and this is Psi5. So this is printed with every single probe, and this channel allows our software to pick up the orientation of the grid in the bottom of each well so that it can perform the computational analysis correctly. For sample collection, we recommend collecting 3 or 4 leaves on the individual plant, ideally focusing on young emerging leaves. We think it's very simple to do this leaf cap punch method. So you will punch a hole in the leaf to collect a couple leaf disc samples for processing. Once you have those leaf discs in the lab, you can put them into a maceration bag. You will grind those up with a grinding buffer, and then you will you will then take a portion of that, 8 microliters, and add that to a denaturing buffer and heat that sample at 95 degrees Celsius for 10 minutes. And then that is ready for your RT-PCR reaction. For our internal validation of this assay, we first, blasted our in silico primers and probes against the different targets and against other targets to make sure we're not crossing with different organisms. We then designed synthetic DNA blocks to confirm that our different probes and primers were functioning properly. Last, we started working with live virus samples obtained from the ATCC collection and other collaborators that we're working with. Some of these are from tomato and different kinds of crops. And finally, we started working with cannabis and hop material. So hops, or Humius lupus, is actually in the same family as Cannabis sativa, so it can be affected by a number of the pathogens that are in cannabis. So it's a nice system to work with when we have difficulty getting cannabis samples. And lastly, we determined the analytical sensitivity and specificity of the assay. Here's an example of the RFU, or relative fluorescent unit output, of individual probes that are specific to the different viruses. In this case, we're looking at beet curly top virus sample from tomato from the ATCC. And you can see that the probe signal is going off quite nicely. And the cannabis beta tubulin signal is not going off because this is not a cannabis sample. Over here, we mixed together a AMV and TMV sample to test if our assay can pick up co-infections. In this case, we mixed an extraction of AMV and TMV together and ran that on the platform, and you can see both our TMV and AMV probes are going off quite nicely, passing the positive thresholds. So this again tells us that we can pick up co-infections as well, because it is possible for your plant to be infected with more than one virus at a time, and multiple virus infections can sometimes have a synergistic or additive effect, meaning that the plant may be even sicker if it has more than one virus infection rather than having a single. In cannabis, this is an example of HLVD positive plant. So the beta tubulin signal is going off. So this tells us that the technician added a cannabis plant to the well. And then the HLVD signal is going off. So this tells us that that cannabis sample that was added is positive for hop latent viroid. And this is a QRT-PCR HLVD assay. So you can see that our positive control is going off here. And that same sample is testing positive for HLVD, our no template control. We get no amplification. For validation, we used a comparator method for all of our different assays. And you can see over here, it was either RT-qPCR assays or an RT-PCR gel-based assay to confirm either negative or positives. And we had 100% agreement with our comparator methods. Most of the samples we had tested were HLVD positive. So again, this is a huge issue in the cannabis market, but we were still able to validate on a number of samples for all of our different virus targets. We have pretty good confidence that it will perform quite well in the field. far as matrix interactions in the analytical LOD, for most of our targets, we were able to get down to 10 copies per reaction and have a positive hit. And this is done with synthetic DNA or RNA transcripts that we generated in-house. And for some of the targets, they had a little bit lower LOD, but still, this should be well above the limit of detection that's needed to pick up these viruses as far as the titers that we've seen in plants that we've tested thus far. We did not see any issues with genomic material from these different crops interacting with our assay, so that was also good to hear. And that's about it for our summary. PhytoX is an RT-PCR microarray-based detection platform. It detects 8 viruses and 1 viroid. It's compatible with a simple crude extraction technique, which means you can extract your samples relatively quickly without needing time-consuming and expensive spin columns. We validated this assay on 240 samples of infected cannabis and other host plants. And our enhanced multiplexing ability goes beyond the limits of a qPCR-based test, which means you will need to order less assays, and that will equate to less cost per plant tested using this platform. And in general, with plant disease diagnostics, it is, again, best to test and not guess. You, again, as we talked about earlier, if you see symptoms on your plant, you don't know if it's a virus, you don't know if it's a nutrition issue. So with viruses, they can become such an issue in your growing operation that if you can determine what the root of the problem is, if it's not a virus, that's great. But if it is, knowing that information will help you hopefully get control and mitigate the impact of the virus in your growing operation. And with that, I just want to say thank you to our customers, and we, are looking forward to developing, other targets that people are interested in. So if you have any questions as far as this assay or how it could be implemented in your lab, just let us know. We'd be happy to talk with you. And we are always open to adding additional targets onto this platform so that we can provide a product that will be useful to both growers and lab operators in the cannabis industry. Thank you.